Hello, good morning to everyone, ladies and gentlemen, doctors and professors. Welcome to our webinar, our highly sought after Back to Basic webinar series. Today we'll be focusing on episode 2, which is the molecular cloning. On top of this, we will have various activities too after the talk. That will be before 12 p.m., just nice before the lunchtime. Before that, allow me to introduce to you this talk, which is brought to you by our collaboration between Thermo Scientific and Apical Scientific. And for the person behind the scene, Mr. Dunstan Lim, Channel Manager of Thermo Scientific, and also our host, Dunstan, please say hi.
Hi, good morning everyone. Thank you for joining us today.
We have as well Dr. Audrey Lim, Product Manager for Thermo Scientific for Southeast Asia. She'll be our main support for Q&A session. Dr. Audrey?
Hi, morning everyone. Yeah, thanks for having me here today.
Yeah. Followed by Ms. Loket Lei, which is our main speaker. She's the Field Application Specialist for Apical Scientific.
Hello, good morning everyone. Welcome.
Last but not least is me. I'm Kevin Koo, Sales Manager of Apical Scientific. So I'll be the co-host and MC for today. Meanwhile, allow me to share as well the housekeeping announcement. So all participants are fully encouraged to join from the beginning until the end of the webinar, and we have prizes to give away later in this session. So the host will actually mute all participants for an enjoyable session. A link to recorded webinar and slides will be sent after the webinar. All participants will be provided e-certificate upon completion. If you look into this app, Cisco WebEx, you'll find that there are a few functions down there on the bottom right side. You may need to explore a bit because it may appear as a chat box button or a 3-dot button. So click those. You will find that chat box button where you can actually text us, say hello to us or anything. If you have questions throughout the event, you can actually drop your question into a Q&A box in those buttons. So at the end of session, we will have a poll box as well for you to help us to give us valuable feedbacks. Okay, so as an example, you may look here to the right side. These are the buttons. So as for the agenda, we have passed the welcome. We will commence the the presentation soon by Ms. Loket Lei, then followed by a Q&A session 11:20, and the most exciting part as well, which is the Menti quiz, where we'll give away prize followed by a poll. So without further ado, we shall start now. Thank you for your attention thus far. I shall pass the floor to Ms. Gatley.
Here, Kevin. Dunstan, can you pass the presentation to me? Okay, so yeah, very good morning to everyone. So welcome to our webinar today. Are you ready to go back to basic in molecular cloning? So let us start from here. In today's agenda, we'll go through an introduction on what is molecular cloning and the basic of it. Then I'll walk you through the various steps of traditional molecular cloning, including how to prepare the DNA insert and vector, followed by the insert analysis, purification, and finally transformation. And I will finish off by introducing to you some of our molecular cloning kit. Okay. So to start off, what is molecular cloning? Here we have a bacterial cell. Okay, so bacterial cells are quite special in that most of their genetic material is contained in a single bacterial chromosome. So it is in gray here. Okay, so this one. Then they also have smaller circular DNA, which we call it as plasmid. Plasmid is not really essential for day-to-day survival, but it contains genes that confer them some properties such as antibiotic resistance. Okay, so over the time, this plasmid it will multiply within the same bacterial cell. Then when the bacterial cell divide, the plasmid are randomly split into each of the daughter cell. And as the daughter cell continue to divide, you will get more and more copy of this plasmid. So, using this property of the bacteria, molecular biologists have the gene of interest, and then they have the plasmid, and we will put the gene of interest into the plasmid to make what we call as recombinant DNA. Okay, so after that, we'll insert this recombinant DNA into the bacteria in a process called transformation. And over the time, we will get many copy of the gene of interest. So this is the molecular cloning in a nutshell. Okay, so again, molecular cloning is actually a set of technique to create recombinant DNA molecule, or we call it recombinant plasmid, okay, which it is actually a simple molecules where you insert a piece or pieces of the DNAs containing the gene of interest into a DNA vector. Okay, so to do this, the DNA to be cloned is first treated with special enzyme to generate the appropriate DNA fragments. The fragments are then combined with the vector to generate the recombinant DNA molecule. And by transforming the recombinant DNA into a bacterial cell, you can quickly generate many copies of the gene. Okay, so now you may ask me, why not just do a PCR? Just recall from our last episode on PCR, DNA polymerase are actually limited at its fidelity and also length of DNA that it can amplify. Okay, so with molecular cloning, the replication of DNA sequence is almost always accurate, and you can copy even a very long sequence. So you can also use cloning to study the function of the gene. But what will happen if I express this gene in the bacteria? I can even take all this plasmid and transfer it into another cell type and see what happened when I express the gene. Let's say in mammalian cell. Okay, so similarly, by expressing the gene, which then get the translate into the protein, actually I can use molecular cloning for protein expression. Okay, so now more basic, let's take a look at some common features of the DNA vector. Okay, for those who are new in cloning, this may look quite complex, so let me break it down for you. Okay, so this is called a vector map. Okay, so first we have the name of the vector here, which usually it will begin with a small alphabet P, and below it is the size of the vector. Okay, so this vector is called PCR vector, and the size is 3.9 kilobase pair. Okay, so next is all vector, they will have origin of replication, which determine the plasmid copy number, or how many copies of the plasmid will there be inside one bacterial cell. Okay, another important features of vector is the antibiotic resistant marker. So downstream, by plating the bacteria on agar containing antibiotics, this will allow you to select the bacteria that contains a plasmid, as those without the plasmid contain the antibiotic resistant marker, they will not survive. Okay. Okay. Next, the important features is the multiple cloning site, which is within the selectable marker. Okay, So in this case, it is the ligase gene here. Okay, so the multiple cloning site, it is basically a sequence that have many recognition site for various restriction enzyme. So that is where we are going to insert our gene of interest. By placing the multiple cloning site within the selectable marker, this will allow me to identify the bacteria that has taken up a recombinant plasmid. So I will share more on selection later on this presentation. Okay, so finally is the multiple cloning site. It usually will flank by promoters if it is for gene expression. Okay, so it is a if it is a gene expression vector, then it will have this. Okay, so Thermo Scientific, we offer a wide range of cloning methods depending on your application needs. Here are some of those that we offer. The most traditional method is restriction enzyme cloning. And on the right here are some and more advanced cloning method. So if you need fast and effortless PCR cloning, Topo technology allows you to clone in just 5 minutes at room temperature. And if you need to shuttle your insert from vector and between species, gateway cloning technology is designed for this. Okay. Okay, next, if you need to clone multiple fragments into a single construct, there are variety of seamless cloning kit available depending on the number of fragments that you want to insert in. Okay, and finally, we can also order custom gene and construct with gene art service. Okay, so of course for today, we'll focus on going back to the basic with traditional restriction enzyme cloning method. Okay, so let's dive in more. Okay. For traditional cloning workflow, it usually begins with restriction enzyme digestion to generate compatible ends between the insert and the vector. Okay, then the DNA end modification, such as dephosphorylation and blunting, may be necessary following the restriction enzyme digestion. Okay, next, the insert and vector are purified by either running on agarose gel and cut the appropriate band and use the spin column method to purify, such as gene jet clone purification kit. Okay, then the insert and vector, they will be ligated to permanently join the insert and vector together. So this is under ligation. Okay, so next, after that, the recombinant plasmid, after the ligation, you will get recombinant plasmid and it will insert into bacterial cell in a process called transformation. And finally, it is the screening and sequence verification will be performed to identify the bacterial cell, whether it have taken up the recombinant plasmid with your desired sequence or not. Okay, so this is the whole workflow for traditional molecular cloning. Okay, so you have known the workflow for molecular cloning. Now we go into more detail on step by step for the workflow. So let's talk about the first step on how to prepare the insert and vector for cloning. Okay, so first of all, how to choose a cloning vector. There are several factors that you need to consider. Okay, so first is the insert site. Most general cloning plasmid, they can actually carry a DNA insert up to around 15 kb in size. Insert larger than that, it may affect the insert stability. Okay, so you may need to get another vector that is better. Okay, then the copy number. Copy number, it refers to the number of copies of the plasmid in each bacterial cell. Okay, in most cases, a high copy number plasmid is desired, but in certain cases where high copy number of the plasmid can become toxic to a cell. So the solution is to use a low copy number plasmid. Okay. Okay, next is the cloning site. Cloning site is important to check the plasmid and selection restriction enzyme are compatible. So as shared previously, most modern vector have an artificial stretch of DNA with many different restriction enzyme cutting site. Otherwise, we also call it as multiple cloning site or MCS. Another one is the selectable marker. Almost all vectors are designed such that only cells that have taken up the plasmid can survive in the presence of specific antibiotic. So this it will actually help to discriminate which cell have incorporated the plasmid. For example, the beta-lactamase gene, which you can see here, it confer resistant to the antibiotic like ampicillin. Okay, and then next is the reporter gene. Reporter gene are used in some cloning vector to facilitate the screening of successful clone by using features of this gene that allow successful clone to be easily identified. Okay, for example, show here is the ligase fragment. Okay, for blue-white selection. Okay, and finally, if your intention is to express the gene, then cloning vector must have the right promoter for transcription. Okay. Okay. So the main player in preparation of end of the vector and the insert are restriction enzyme. In short, we call it RE. Okay. Sorry. So RE also called as restriction endonuclease. They are able to find and cut a specific recognition site in double-stranded DNA. So these enzymes are used in order to prepare the vector and insert DNA to have complementary end that can be ligate together. So depending on where exactly the enzyme cut in its recognition site, the resulting product, it will either have a cohesive end that are commonly also we call it as sticky end, leaving this type of overhang of 1 to several nucleotides, usually up to 8, or a blunt end like this. Okay, so blunt end, it must be treated in order to ligate to another DNA molecule. While sticky end, it can be reattached to other DNA with matching ends. Okay. Okay, here are some common restriction enzyme cloning strategy. Okay, first, it is the cloning with 2 sticky end. For this, the vector and insert end need to be compatible. If done correctly, your vector and insert ligation is efficient. So vector self-ligation will be low, and it will ligated cleavage site are intact. Okay, so this cloning is directional, which means you know where is the direction of your insert, as there is only one possible way for the insert to join so to the vector. So it is using the complementary sticky end. Okay, then the second strategy is cloning with 2 different and compatible ends, such as XHO1 and SAL1. here. Okay, so they are same with the first strategy, but the cleavage site of the original RE may be destroyed after ligation. Okay, the third one, it is the cloning with one sticky end and one blunt end. So it also a directional cloning. However, ligation of the blunt end may be less efficient. Okay, the last one is cloning with blunt ends. So this is not a directional cloning. So you will have to screen the clone to ensure you have the correct sequence orientation. And modification may be required after restriction enzyme digestion to ensure compatible blunt end. With blunt end cloning, the ligation will be less efficient than sticky end cloning, and vector recirculation will be more higher. Okay, so ligation of two DNA fragment, for example, vector and insert, they actually need at least one of the DNA ends to have a 5' phosphate for the nucleophilic attack of the 3' OH hydroxyl group. Okay, to end with a phosphate group. Okay. So, conventionally, restriction enzyme, they will typically will take up to 1 hour or overnight to complete the DNA digestion reaction. Okay, so different RE will require different restriction buffer. As such, when you perform multiple digestion using 2 or more RE in the same reaction, then you will need to have optimized the buffer. This may be a quite tedious process. Okay, so because of the long digestion time, and also there is also risk of star activity, which refer to a non-specific cleavage of the DNA outside the restriction site. Okay. Okay, so here is just to share. Here is the Thermo Scientific history of restriction enzyme development. In 1977, we launched a screening and characterization program. In this program, our scientists collect samples from various locations, such as soil, water, and so on, and take them back to the lab to grow the bacteria from this sample. Okay. From there, they screen for any interesting new and unknown restriction enzyme that they could isolate and use in the lab. So here in the picture, we have two of our scientists. They are collecting sample in Ukraine and around some of the hot spring in also Ireland. Okay, so through this screening program, more than 30% of all restriction enzymes known today were discovered by our scientists. Okay, so this was how we generate a large collection of 200 commercially available restriction enzyme. And on top of that, as I mentioned in a previous slide, one challenge of conventional restriction enzyme is the multiple buffer system. So we were the first to introduce a new standard, the one buffer system, to address this challenge. Okay? So here, one elegant option to avoid all the incompatibility in buffer, the extended incubation and star activity, is by using Thermo Scientific fast digest enzyme instead of using conventional restriction enzyme. Okay, so fast digest RE are a cut above the conventional restriction enzyme in that it is super fast. It can complete most restriction digestion within 5 to 15 minutes. Okay, and on top of that, all 176 fast digestion enzyme using a universal buffer system, okay, which it make it easy for you to do multiple digestion, as there is no need for buffer optimization. The buffer is also 100% compatible with downstream application. Okay, so fast digest enzyme come in either colorless or with green buffer format, which it contain a loading die for direct gel loading after restriction digestion. So the green buffer, it will separate into blue and yellow band, which migrate at different speed upon the gel electrophoresis. So it gives you the visual cue on how far your band have resolved in the gel electrophoresis. Okay. Okay. Now let's take a look at fast digest restriction enzyme in more detail. In the gel image here, okay, you can see the comparison of digestion time between fast digest and conventional restriction enzyme and its performance. Okay, so with fast digest, it complete the digestion in just 5 minutes for double digestion, means for XHO1 and APA1 here. Okay, so when you combine for double digestion, the total digestion time is also 5 minutes only. Okay, so in contrast with conventional restriction enzyme, it will take 60 minutes for single digest. Okay, and for double digestion, it will double the time, which it actually needs 120 minutes here. Okay, because if you look at this workflow, for conventional restriction enzyme, you will have to do the digestion sequentially, whereas for fast digest, you can actually do the multiple digestion all in the same buffer, because all 176 fast digest enzyme are 100% active in the same buffer. Okay, so therefore, fast digest restriction enzyme really will save your time and especially convenience when you have many samples. Okay. Okay, so by using fast digest also, it can actually help to take the guessing game out, because one unit is normally defined as the amount of enzyme required to digest 1 microgram DNA in 1 hour in 15 microliter, but this is actually typically tested on lambda DNA in an optimal buffer only. Okay, so what if you have PCR product, it's not a lambda DNA or plasmid or gDNA, and what if the reaction is not in an optimum condition? Okay, so with fast digest, the recommended digestion time are provided for all type of DNA template. And unlike unlike for conventional restriction enzyme with fast digest, no overnight digestion are required for any template, and eliminating the need for prolonged incubation time in turn eliminates the risk of the star activity also. Okay. Okay. So as mentioned previously, fast digest RE are 100% compatible with the downstream application, such as DNA and modification. Okay, so here we show that all the typical and modifying enzyme used for DNA blunting, vector dephosphorylation, and DNA ligation, they are 100% active in the fast digest buffer. Okay. Okay, and here are some resource we have for fast digest restriction enzyme. we have an online selection tool which allows you to search by product name or isochizomer name, recognition site, or SKU number. Also, we have application notes on multiple digestion, scaling up digestion, and also so on. So for more knowledge, we also have some article such as common issue with restriction digestion reaction, and also how to avoid them. Okay, so do take a look at our website. Okay, so before we move on to the next topic, here are some tips for preparing your insert. Okay, first, if your insert is a PCR product, you can design your primer with the appropriate RE site so that it can digest with the restriction enzyme. Okay. Secondly, when the cloning for applications such as gene expression or sequencing or SNP analysis and so on, it is important to use a high fidelity polymerase such as our latest fusion DNA polymerase, which have more than 100% sequence accuracy as compared to regular tag DNA polymerase. Okay, to generate a blunt end PCR product, a high fidelity proofreading polymerase can also use, whereas a regular tag polymerase, it will add an extra DA untemplate overhang at the 3' end. Okay, so do take note on the polymerase that you are using. Okay, so now that we have already covered the topic of restriction enzyme, let's move on to the end modification that are performed after the restriction digestion. Okay, so for blunt end cloning, you will have sometime need to generate a blunt end cloning from a sticky end. Okay, so blunt end, it can actually generate by filling in the 5' overhang or chewing back from the 3' overhang. Okay, both T4 DNA polymerase and kernel fragment, they can fill in the 5' overhang, and the DNA, T4 DNA polymerase, it also possesses the 3' to 5' exonuclease activity, which it can chew back the 3' overhang. Okay, so usually a combination of T4 DNA polymerase and kernel fragment is used for blunting. Okay, okay. Next, if you have made a single cut in the vector, which common for blunt end cloning, it is easy for the vector to recirculate. Okay, so meaning the ring, it can be easily closed back again without any insert inside. Okay, so this phosphorylation of the vector DNA is used to prevent the vector recirculation during the downstream ligation reaction. Okay, so you can perform the phosphorylation by using Thermo Scientific fast AP thermo sensitive alkaline phosphatase. Okay, the reaction with this enzyme, it can take only 10 minutes at 37 degrees Celsius, and one protocol, it can apply for all types of the DNA ends. Okay, so for ligation of your insert and vector to occur, at least one of the DNA end that is either insert or the vector, they will need to contain a 5' phosphate. Okay, a phosphate at the 5'. So in the slide before this that I mentioned just now, I've talked about the phosphorylation of the vector to prevent recirculation, right? So if your vector already is dephosphorylated, okay, that means take out the phosphate, then your insert must have a 5' phosphate for the ligation to happen. Okay, so if you are preparing your insert using restriction enzyme, then there is no problem because RE digestion, it will always produce N with 5' phosphate. However, very often, especially for blunt end cloning, your insert is a PCR product. Okay. PCR product, it usually do not contain a 5' phosphate unless you design your primer that you'll be used in your PCR to have a phospho-related primer. Okay, so if you are using a normal primer, then you will certainly need to generate a 5' phosphate to the insert to the PCR product. Okay, so for this, you can consider using Thermo Scientific T4 polynucleotide kinase, or in short, we call it T4PNK for this purpose. Okay. Okay, so we have talked about blunting using T4 DNA polymerase and Cranal fragment, and we have also talked about phosphorylation using the T4 PNK. So this brings me to our Thermo Scientific Fast DNA and Repair Kit, which it is efficiently perform the blunting and also the phosphorylation for up to 5 microgram of the DNA in just 5 minutes. Okay, so this kit, it contains T4PNK, T4 DNA polymerase, and the Cranal fragment in a convenient enzyme mix format, okay, to reduce all the pipetting steps. Okay, so here is the summary of what I have talked so far. For blunting, we have the Thermo Scientific Cranal fragment and Thermo Scientific T4 DNA polymerase. To prevent vector recirculation, we have the Thermo Scientific Fast AP thermosensitive alkaline phosphatase, which it dephosphorylate all type of DNA ends in just 10 minutes at 30 degrees Celsius. And then for ligation to happen, at least one of the DNA end, it will need a 5' phosphate group. So Thermo Scientific T4 polynucleotide kinase, it will help you on this. Okay, and finally, for convenient and efficient blunting and also phosphorylation, we have Thermo Scientific Fast DNA and Repair Kit, okay, which is an enzyme mix containing the Cranal fragment, T4 DNA polymerase, and T4 PNK all in one. So it does the blunting and phosphorylation in just 5 minutes. Okay. Okay, now let's move on. After the preparation of your insert and vector, we move on to the next step, which is the analyze and purify of your insert. Okay, so after restriction enzyme digestion, the next step is to perform a simple gel electrophoresis for your digested product to select the correct fragment based on the size. Okay, do not forget on our previous episode 1 in Thermo Scientific, we offer a complete range of reagent for DNA gel electrophoresis, including our gene ruler DNA ladder, our tri-track loading die, and also as well as our top vision agarose. Okay, so we have a complete range of the reagent. Okay, so after you have identified your DNA fragment of interest that you want, cut out the band from the gel and isolate the DNA from the gel. Okay, so for this, you can use gel extraction kit, such as our Thermo Scientific GeneJet gel extraction kit. Okay. Okay, so next is ligation. In ligation, okay, ligation is actually the formation of covalent bonds between the 3' hydroxyl group and the 5' phosphate of the two DNA fragments. So it fusing the two fragments together. Okay, so this reaction is catalyzed by Thermo Scientific T4 DNA ligase. Okay, so for ligation, the preparation of the ligation reaction is very important. Here are some tips for you. Okay, firstly, you can try to titrating the molar ratio of vector to insert from 1 to 1 to 15 to 1. Okay, usually an excess of insert will work better. Okay, so most conventional ligation can be complete in 1 hour, but sometimes increasing the ligation time, it can also help. Okay, so you can also try to perform the ligation at 16 degrees Celsius for overnight. Okay. As I mentioned just now, conventional ligation, usually it will take at least 1 hour to complete or overnight. Okay, you can actually use the more scientific rapid DNA ligation kit. It can enable fast sticky end or also blunt end DNA ligation, and it is performed within 5 minutes at room temperature only. Okay, so the kit, it actually contains T4 DNA ligase and also a specially formulated rapid ligation buffer. So it is optimized for fast and efficient DNA ligation at the room temperature. Okay, so fast ligation efficient is equal to the obtained with T4 DNA ligase in a standard 1 hour ligation. Then the ligation reaction mix can be used direct for bacterial transformation. Okay, okay, here do note, however, for electroporation, when you perform the transformation, the T4 DNA ligase will need to be inactivated before you use it. Okay. Okay, so now let's move on to the next step. So after ligation, you get the recombinant plasmid. So the next step is the transformation. Okay, here are some commonly used host cell for cloning, in particular for protein expression. Okay, so bacteria is actually commonly used, the most commonly used, because it has the fastest and most efficient growth. Okay, so it is scalable and is low cost with the simple culture condition only. Okay, but however, for bacterial cell, it can get challenging when trying to express mammalian protein, and there are issues with protein folding and solubility. Okay, so next is yeast cell. Okay, yeast cell, they are scalable, have a simple medium requirement, and they have eukaryotic protein processing mechanism. However, yeast cell, the growth condition may require optimization, and fermentation is needed for a very high protein yield. Okay, so next is the insect cell. In insect cell, they are actually popular choice for protein expression using the baculovirus expression system, because these cells, they are able to express large quantity of protein with complex post-translation modification. Okay, but however, the culture conditions are more demanding, and the production of the recombinant baculovirus vector may take times. Okay, so most ideal expression system for protein expression is, of course, the mammalian cell, because due to its highest level of protein processing. However, this cell, it require more specialized culture condition, and higher protein yield will require suspension culture. Okay, so today we will focus on the bacterial cell, which is most commonly used in the lab to propagate clone plasmid. Okay, the process of having a bacterial cell to update your recombinant plasmid, it is called the transformation. Okay, so for transformation, we use a special type of bacterial cell called competent cell. Okay, so this cell, they have been made porous for efficient transformation. Competent cells are available commercially, and this usually give the highest transformation efficiency, or they can also be made competent in labs. Okay, if you want to make it in lab, there is two types of competent cell. Okay, so chemically competent cell are calcium chloride treated to facilitate the attachment of the plasmid DNA to the cell membrane. Okay, to get the plasmid into the cell are heat shocked at 42 degrees Celsius for 30 seconds, and this opens the pores of the cell membrane so that the plasmid DNA can enter the cell. Okay. Next one is the electrocompetent cell. They are prepared for transformation using a short electric pulse from the electroporator. Okay, so this electric shock, it will create pulse so that the plasmid DNA can enter into the cell. Okay, so for chemically transformation, it is the most commonly used method for the application, like routine cloning and protein expression application, because it is well established and does not require any special equipment. Okay, while for more challenging application, such as you're trying to construct a cDNA or gDNA libraries of various DNA size, or you are going to do a transformation for a few low quantity of plasmid, like in a picogram range, or you are trying to transform a very large DNA above 30 kb, then electroporation is more preferred. Okay, electroporation, it require an electroporator which limit its scalability, and it will require some protocol optimization and also some skill. Okay, however, electroporation will give significantly better transformation efficiency than the chemical transformation. Okay, so electroporation is also compatible with a wide range of bacterial species, including those with cell walls. Okay, just to share, I tried both this before. Electroporation, if you success, then you can really get a lot of the bacterial cell. But if you fail, it's quite difficult. You will get a plain plate. Okay, so more easier is to get a commercial one. Okay, so in Thermo Fisher Scientific, we offer a wide range of both chemically and also electrocompetent cell. Okay, so we have four types of the chemically cell, which are most commonly used in the lab every day. For cloning of a large plasmid or site-directed mutagenesis, we have DH10B competent cell. And for routine cloning, we have the DH5A competent cell with 2 transformation efficiency standard and sub-cloning efficiency. Okay, and finally, for protein expression, we have the BL21DE3 strain. Okay, and we also offer this competent cell as a combination kit with our CloneJet cloning kit, or the fusion site-directed mutagenesis kit. Okay, I will touch on the CloneJet cloning kit later in a very shortly. Okay, so as I mentioned, commercially available competent cell, it will give the best transformation efficiency. However, if your budget is really constrained, there is always a way to make your own competent cell. Okay, so you can consider Thermo Scientific Transform 8 Bacterial Transformation Kit. It will allow you to do the competent cell by your own using a simple kit with a fast protocol. It takes only 1 hour from overnight bacterial culture to cell plating and just 2.5 hours from bacterial cloning. Okay, with Transform 8, you can expect better efficiency than traditional homebrew methods with more than 10 to 7 transformers per microgram of the plasmid DNA. Okay. Okay. And finally here, let me share some tips on transformation with you. Okay, so first, do remember it is important to avoid freeze-thaw cycle for the competent cell. As each freeze-thaw cycle, they can lower transformation efficiency by half. This is why Thermo Scientific competent cell, they come in 2 reactions per valve to minimize the need for freeze-thaw cycle. Okay, and another tips is to improve the cell viability and cloning efficiency. It is to culture the transformed cell in antibiotic-free liquid medium for a short recovering period, like for example 45 minutes to 1 hour before plating on the antibiotic medium. Okay, so this will actually allow the time for the antibiotic resistant gene from the acquired plasmid to be expressed first. And we recommend you to use the SOC medium, which can increase the number of transformed colony by 2 to 3-fold compared to LB broth. Okay. Okay, so I've covered the entire cloning workflow now. So let's talk about the cloning kit, which it will give you the all-in-one convenience. Okay. Okay, so before I go on, let me touch a bit on a very important topic on selection. Okay, selection, it is typically performed after the transformation. So selection, it actually will determine whether the colony that you observe after the transformation, is it carry the your gene of interest or not. Okay, so traditionally, selection is performed by blue-white screening. Okay, how this blue-white screening work? If you look at the plasmid here on the left, it has the BLA gene, okay, which it actually code for ampicillin resistant. Okay, so this means that when placed on the agar containing antibiotics, only the cell that have picked up the plasmid, they will survive. Okay, so of all the cell that is survive on your plate, which means there are 2 possibilities, right? One is it may contain the insert in the cloning site, or it may not contain the insert, which means the plasmid has recircularized. Okay, so how do we differentiate these 2? Okay, so in blue-white cloning, blue-white screening, the cloning site in the vector is actually within the ligase gene, which code for beta-galactosidase. Okay, so if the vector recircularizes, the ligase gene, it will express and it will hydrolyze the X-scale to a blue pigment. So this, it will appear as a blue colonies on the plate. Okay, so if there is no insert, so if there is insert, the ligase gene, it will be actually disrupted. and it cannot be expressed. So this will appear as a white colony. So which means when you pick for the colony, you need to choose the white colony for the blue-white screening. Then downstream, you may perform a further analysis such as sequencing to ensure the sequence of the recombinant plasmid is correct. Okay, so now that is the blue-white screening. Now I want to introduce to you an alternative method for screening that it takes much less effort and it is more economical. Okay, so our Thermo Scientific ChronJet cloning kit, it actually uses a positive selection through a legal DNA to a legal gene instead of the expensive blue-white screening. Okay, so this kit, it will give you a high efficiency and given a greater than around 99% the positive clone. So in the next slide, I will share with you more on the positive selection. Okay, so ChronJet kit, it is a all-in-one kit which come with a PJet 1.2 blunt cloning vector, 34 DNA ligase, the restriction buffer, as well as the DNA blunting enzyme. So ChronJet is a versatile kit. It has a multiple cloning site that is compatible with blunt or sticky end cloning, and you can actually complete the cloning in just 5 to 10 minutes. Okay, so it's like 5 minutes for ligation, and the extra 5 minutes is for DNA blunting if it is required. Okay, so it is very fast reaction. Okay, so here is how the positive selection work in ChronJet. The PJet 1.2 blunt vector used in this kit, this one is the vector map. So you can see here, it carry the usual features of the cloning vector. But unlike blue-white screening, where the cloning site is within the ligase gene, the ChronJet, the multiple cloning site, it is within a legal ECO471R gene. Okay, so it means when the vector recircularize, it will express this legal restriction enzyme gene, which it actually will kill the host bacteria after the transformation. Okay, therefore, all the colonies that survive on the plate, they shall contain the recombinant plasmid. Okay, means it will carry the insert. Okay, so there is no need for the blue-white selection that need extra additional cost where you need to buy Xscale, IPTG, or that extra reagent. Okay. Okay. There is another cloning kit that I want to share with you today. This is slightly different technology from what we have talked about the traditional cloning just now. Okay, in Thermo Scientific, it is the Thermo Scientific alligator cloning kit. It is for bacterial protein expression system that uses ligation independent cloning. Okay, it is designed for a fast and efficient LIC cloning and subsequent tight regulation of the gene expression in the E. coli. Okay, first let's talk about what is the ligation independent cloning, LIC. Okay, so as the name, it is LIC. So LIC, it is the insertion of the PCR product into a target vector without any use of ligase or other enzyme. Instead, how it works is by having this long complementary sticky overhang on the insert and the target vector. Okay, here. So after annealing, the LIC vector and insert, they can be directly transformed into the competent cell, and the covalent bond formation at the vector insert junction occur within the cell to yield the circular plasmid. Okay, so there are various advantages on this LIC method. First, it is extremely efficient with low background due to the long complementary overhang, and it also eliminates the variable associated with ligation, meaning less optimization for you. Like I mentioned just now, for ligation, you need to optimize the ratio of insert vector. Okay, so this it less the optimization for you, and also it does not require restriction enzyme digestion prior to the cloning. Okay. Okay, so how this allocator work? Okay, first, the gene of interest, it will be amplified using a PCR primer designed with some extra sequence as specified by the kit. So you need to refer to the kit manual before you design for the PCR primer. Okay, so this it will help you to incorporate this extra sequence to the gene of interest. Okay, so next, the T4 DNA polymerase, it is used to create specific 14 to 21 nucleotide of a single-stranded overhang on the vector and also the DNA insert. Okay, so recall on the T4 DNA polymerase, it has two enzymatic activity. One is 5' to 3' polymerase activity, and another one is 3' to 5' exonuclease activity. Okay, so this exonuclease activity, it will remove the nucleotide from the 3' ends of the DNA, while the polymerase activity, it will restore the chain using DNTPs and the complementary DNA strand as a template. Okay, so the polymerase activity, it will need the presence of the NTP to activate the activity. Okay, so in this LIC protocol, only the GTP will be included in the reaction. So it will causing the 3' to 5' exonuclease activity, and then the 5' to 3' polymerase activity will equilibrate at the first occurrence of the cytosine in the complementary strand, which means when there is a C in the template, then the DGTP will have the action, so it will help to put in the DGTP, so it will have the polymerase activity at this to restore the DGTP here. Okay, so with this, it will generate a long complementary sticky end here, which end with a G. Okay. Okay, so the allocator system, it consists of 4 kits and 2 set based on the pellet, the vector series of the bacterial expression vector. Okay, so for protein with a known preference for either the N-terminal or C-terminal his tag position, using the appropriate N or C-terminal kit is recommended. Okay, so it means if you know the his tag position is at N or C, then you can use the N or C terminal kit. Okay, so when the protein structure and features are not well known, it is recommended to use an allocator set. Okay, the set, it will actually allow the cloning into 3 different vectors to determine the most compatible vector for further experiment. Okay, so the pellet bacterial expression vector are designed for high level of target protein expression in concert with minimized basal expression by having the gene of interest driven by a strong T7 promoter. Okay, so do note that E. coli strain such as BL21DE3, whereas the T7 RNA polymerase gene is under the control of an inducible promoter must be used. Okay. Okay. So following the protein affinity purification, amino terminal tag can be removed either by the enterokinase or also using a novel protease, the well key. Okay, protease. Okay, here, to summarize today's presentation, I have walked you through the basic of cloning workflow from DNA preparation to cloning and finally to transformation and clone analysis. Okay, so we have products in Thermo Scientific molecular biology line to support you in every of these steps. Okay, for insert preparation by PCR, we have our Fusion Plus high fidelity DNA polymerase. We also have PCR plastic. We have nuclease, free water, and DNTP, and also so on. Okay, you can inquire from us. Okay, for cloning, we have the fast digest restriction enzyme. We have various modifying enzyme as well as the cloning kit, such as our CloneJet and alligator cloning kit, which give you all the convenience. Okay, so for transformation, we have our Thermo Scientific line of cost-effective yet high efficient competent cell. Okay, and finally for analysis by gel electrophoresis, we have our gene ruler DNA ladder, we have top vision agarose, we have buffer, and also so on. Okay. Okay, so with that, I come to the end of my presentation. Thank you all. Okay, so we are happy to take up a question now. We have Dr. Audrey to help in answering all your questions. So that's the end of our section of presentation.